Hi, I'm Stema, and I'm a stem cell. Stem cells, like me, are cells that can, number one, make copies of themselves indefinitely under the right conditions, and two, specialize into more specific cell types, like eye or liver cells. Right now, other stem cells in eye are being used to treat many diseases, and stem cell treatments are in trials for countless other diseases. The most common and only approved stem cell treatment in many countries is a blood stem cell or bone marrow transplant. Bone marrow transplants are used in quite a few instances, including when normal blood cell forming cells are damaged due to chemotherapy for cancer patients. Other therapies undergoing clinical trials include a treatment for advanced Parkinson's disease, in which stem cells are specialized or differentiated into the specific type of neuron that's damaged or lost in Parkinson's disease, and then those cells are injected into a specific part of the brain. In another trial, patients with severe type 1 diabetes are being infused with insulin-producing islet cells, which initially come from, you guessed it, stem cells. It's thought that the first patient in this trial has been essentially cured of type 1 diabetes. As you can see, stem cells like me are helping with many diseases and lots of research right now, but my story starts quite a bit ago. So, here is the story of my life. Stem cells have been around in the bodies of animals for many, many years, but their existence was largely unknown until 1961, when James Till and Ernest McCullough, both of whom received Canada Gairdner International Awards in 1969, discovered the first stem cells. The cells they discovered were blood stem cells. These cells were the ones we've already talked about that are now being used to save many lives each year. Blood stem cells are able to specialize or differentiate into any of our blood cells, such as red blood cells, platelets, or white blood cells. The next groundbreaking stem cell advance came in 1981, when Martin Evans and Matthew Kaufman cultured the first mouse embryonic stem cells, which means they grew them in a dish in a lab. Embryonic stem cells are pluripotent, and pluripotent stem cells are a specific type of stem cell that can specialize into any body cell, like heart or liver or blood cells, but not structures outside of the embryo, like the placenta. Think of pluripotent stem cells as children. Young kids are often still deciding between many careers for their future, but they may have eliminated some careers from their list based on their life experiences. Skipping ahead by about a decade, we reach another discovery about embryonic stem cells led by Janet Rossent, who is the president and scientific director at the Gairdner Foundation, and Andras Nagy. Starting with a paper in 1990, Drs. Rossent and Nagy and colleagues began testing mouse embryonic stem cells' pluripotency. Their experiments weren't fully successful in 1990 in proving this, but in 1993 they did it. Boom! Pluripotency proven. Just a year later, in 1994, another seminal paper was published, this time about cancer stem cells, by Tzvi Lapidot, John Dick, who's received the Canada Gairdner International Award this year in 2022, and their colleagues. Cancer stem cells are, quote, cancer cells present in cancer tissue that have changed their appearance such that they resemble and behave as stem cells, end quote. It's likely that cancer stem cells are largely responsible for spreading cancer. But what do these cells mean for treatments? Lots of different things, as it turns out, depending on the type of cancer. For one type of leukemia, called acute promyelocytic leukemia, or APL, scientists induce the specialization of the cancer cells, and this helps make chemotherapy safer. In other cases, the signals that cells receive from their environment can, quote-unquote, create cancer stem cells. So studying the pathways and signals that lead to this gaining of stemness and finding ways to eliminate those may lead to new treatments. Let's fast forward another 4 years to 1998. It's nearly the turn of the century, and another amazing discovery is around the corner. James Thomson and colleagues have isolated human embryonic stem cells for the first time. The importance? In the words of the authors of the 1998 paper, the standardized production of large, purified populations of human cells, such as heart cells and neurons, will provide a potentially limitless source of cells for drug discovery and transplantation therapies. Our penultimate important event comes in 2006, when Kazutoshi Takahashi and Shinya Yamanaka, recipient of the Canada Gairdner International Award in 2009 and later a Nobel laureate, showed they could turn mouse fibroblasts, which are connective tissue cells, back into pluripotent stem cells. A few decades prior to this, it was thought that stem cells could specialize, but when they became those specialized cells, like an eye cell for example, they could never go back to being a stem cell. That idea had been turned on its head over the years, but this discovery conclusively showed this wasn't true. Takahashi and Yamanaka's cells are called induced pluripotent stem cells. 1 year later, in 2007, they showed the same process of making specialized cells into stem cells was possible for human cells too. This has allowed for the creation of patient and disease-specific stem cells. The cells are now being used to create therapies where we can replace a patient's damaged cells with new cells that match them genetically. Finally, in 2009, another seminal paper was published by Toshiro Sato, Hans Clavers, and colleagues, demonstrating the ability to grow organoids, which are, quote, 3D structures grown from stem cells that consist of organ-specific cell types that self-organize, end quote. What does self-organize mean? It means the researchers don't need to tell the cells where to go or how to create the organoid. The cells do that on their own, mimicking normal development when in the right conditions. While they operate completely differently biologically, it may help to think of this idea as a flower. You don't need to tell a flower how to grow. It does that on its own as long as it's provided with water and sunlight. Since then, scientists have cultured organoids recapitulating many tissues, such as the liver, pancreas, kidney, prostate, lung, brain, and many others. We now even have organoids of lacrimal glands, which are responsible for making us cry or producing tears. These organoids can actually cry too. Organoids are great because they better mimic what goes on in our body. Traditional 2D cultures are layers of cells, not really like how the body operates in many instances. Organoids also provide a way to check if a certain drug will help a specific patient, as we can use stem cells from that patient. So, that's the story of my life. The list of important events wasn't nearly exhaustive, but we've covered some critical discoveries. There are also some newer, super exciting events we didn't get a chance to explore. Still, it's clear I've had quite the adventurous life, but there's still lots more to come. I hope you choose to follow along.